Metabolism.
Okay. So today, for the next three classes, 2 to 3 classes, we'll be discussing about cholesterol metabolism. Okay. So in today's class, there is some mix-up in the. So for the next three classes, we will be discussing uh, cholesterol metabolism. That is, its biosynthesis and breakdown and its utilization. So the metabolized products, utilization of catabolized products. Okay. So in today's class, we will be discussing about biosynthesis of cholesterol. Okay, what are the objectives of today's class? At the end of this class, you people will be able to recall different stages of cholesterol biosynthesis, explain reactions involved in different stages of cholesterol biosynthesis, explain regulations of cholesterol biosynthesis, regulation of cholesterol biosynthesis. Okay, so these are the three major objectives. So before we get into those objectives, we will try to know what is cholesterol. Cholesterol is an steroid containing molecule. In its chemical structure, it contains steroid. So cholesterol is a steroid containing molecule. In its chemical structure, it contains steroid. So this is the steroid skeleton. So we will see some of the features of steroid. So steroid is a biologically active organic compound with 4 rings arranged in a specific molecular configuration. Okay, so this typically it is composed of 17 carbon atoms. So you can look into here, the numbering starts from the crown carbon of a ring A. So here it contains 4 rings and they are named as A, B, C and D. It is always in a steroid nucleus. These rings are referred in an identical manner: A, B, C and D. The numbering starts from the crown carbon of A. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 and 17. So the basic steroid skeleton consists of 17 carbon atoms bonded in 4 fused rings. So 3 6-membered cyclohexane rings. So the A, B and C consists of 3 6-membered cyclohexane ring and 1 5-membered cyclopentane ring. So this is the composition of steroid nucleus. If you want to call a molecule or a biomolecule as a steroid, it should contain this skeleton. So steroids and their metabolites function as signaling molecules, like for example steroid hormones. So they help in signaling, that is, they pass on the information from nervous system to brain. Okay, steroids and phospholipids are components of cell membrane. So they are one of the important components of cell membrane. So steroids such as cholesterol, so these cholesterol decreases the fluidity of the membrane, so it increases the sturdiness of the cell membrane. Cholesterol, they are highly concentrated energy stores, so they have a lot of energy in them, but they are not typically utilized as a source of energy in mammals. They are normally metabolized and excreted. How they are metabolized and excreted, that we will see in your future classes. Okay, steroids in animals are derived from lanosterol. Lanosterol is the basic sterol that through which steroids are further synthesized, and in plants it is a cycloartinol. Okay, so the only difference in its chemical structure is the chemical structure. If you can compare the chemical structure of lanosterol and cycloartinol, so here at this carbon atom, so that is at the 6th carbon atom, so you can see there is a cyclization in the cycloartinol. Okay, So that is the major difference between lanosterol and cycloartinol. So sterol of mammals, and this is the sterol of okay. So these sterols are the steroids carrying a hydroxyl group at third carbon atom. Okay, so these sterols means what? They are steroids which contain an alcoholic group that is at a particular position, that is at the third carbon atom. If they are containing an hydroxyl group at the third carbon atom, they are called as sterols. Steroids are called as sterols. Additional carbon atoms may be present in the side chain, and the sterol skeleton has a molecular formula of, that is basic sterol skeleton is having the molecular formula of C7H28O. It contains one oxygen and one oxygen in excess. Okay, so now about cholesterol. So cholesterol is the most abundant sterol in human and performs a number of essential functions. So this is the major constituent of plasma membrane and of plasma lipoprotein. So it is the precursor of bile salts. So the role of bile salts that we will discuss in your future class. So precursor of steroid hormones, adrenocortical hormones, and sex hormones. So these are derived, and many more hormones are derived from cholesterol. Okay, precursor, it is the precursor for vitamin D and cardiac glycosides. These are also derived from cholesterol. So it is required for the nerve transmission. Okay, it is required for the nerve transmission, that is transmission of impulses. So the structure of cholesterol. So this is the sterol molecule, and this is cholesterol molecule. So you can look at the difference. So it is having a double bond over here at the 5th and 6th carbon atom. Okay, and at the 17th carbon atom it is having a side chain. How many carbons? 1, 2, 3, 4, 5, 6, 7, 8 carbon containing side chain. It consists of sterol nucleus, an 8 numbered and branched hydrocarbon chain is attached to the D ring at C17. It contains 2 angular methyl groups at C19 and C18. So here and here, so which are referred to as C18, C19 and C18. C19 methyl group is attached to carbon 10 and C18 methyl group is attached to carbon 13. Okay. So the C18 and C19 methyl groups of cholesterol, so they lie above the plane of the 4 steroid basic nucleus, and much of the plasma cholesterol always exists in the esterified form with the fatty acid attached at the third carbon atom. So here you have an alcoholic group, and carboxylic acid can form an ester bond with it. Usually fatty acid form an ester bond, and so they are, when a fatty acid combines with an cholesterol, then that molecule is called as polysteryl ester. These polysteryl esters are more hydrophobic than cholesterol. Okay, so in a lipid membrane, in our cell membrane, so they exist as polysteryl esters. So they they help to make the cell membrane hydrophobic. So now the pathway, how the cholesterol is synthesized inside our body, the biosynthesis of cholesterol. In humans and other animals, the biosynthesis of steroid follows mevalonate pathway. So we are discussing mevalonate pathway. So the synthesis of cholesterol proceeds through the formation of mevalonic acid. So that is why we call it as mevalonate pathway. So here, so the starting molecule is acetyl-CoA. So a 2 carbon containing molecule. So we have been discussing about acetyl-CoA since very long, from many previous classes. So again, this acts as a building block for isoprenoid units. We will look into those isoprenoid units. What are those isoprenoid units? Okay, so that is dimethyl allyl phosphate and isopentenyl pyrophosphate. So again, so these two, dimethyl allyl phosphate and isopentenyl pyrophosphate, combine and they will form 15 carbon unit. Again that will combine and it will form 30 carbon unit. Okay, so that is in subsequent steps, that is these isoprenoid units will join to form geranyl pyrophosphate 10 carbon unit. Again this combines to form 15 carbon unit. Again that will combine to form 30 carbon unit, the lanosterol, which is further converted to cholesterol. Okay, so we look in detail about this. Okay, so before that, about 1 gram of cholesterol is synthesized per day in adults, and almost all the tissues of the body participate in cholesterol biosynthesis. Okay, so but the largest contribution of cholesterol is from liver, that is around 50% of the cholesterol is synthesized from liver and 15% is synthesized from intestine, and small amount is also synthesized from almost all parts of the body like skin, adrenal cortex, reproductive tissues, etc. Okay, so the enzymes involved in cholesterol synthesis are found in the cytosol and microsomal fractions of the cell. So cholesterol synthesis happens in the cytoplasm. So here acetate of acetyl-CoA provides all the carbons of cholesterol. So this is the only thing. Acetyl-CoA is the only source of cholesterol, that is only source of atoms for cholesterol. Okay, so the reducing equivalents are supplied by NADPH and ATP will provide energy. Okay, so this is an anabolic reaction. So we are building large molecule from small molecule, acetyl-CoA. We are taking acetyl-CoA 2 carbon unit and we are building in 27 unit large molecule. So it is an anabolic pathway. So for the production, so it consumes energy. So for the production of 1 mole of cholesterol, you require 18 moles of acetyl-CoA and 36 moles of ATP and 16 moles of NADH. You can imagine how 1 molecule of cholesterol means how much of energy does it store. Okay, so that is why it is called a large energy reserve, but it is not utilized by our body. Okay, so there are 5 stages, but it is having many other significant functions in our body that we discussed in your previous slides. There are 5 stages in biosynthesis of cholesterol. So to make it easier, we have divided into 5 major parts. That is 5 stages. The first stage is synthesis of HMG-CoA. Then the second stage is formation of mevalonate, 6 carbon unit. Then production of isoprenoid unit, 5 carbon units of hydrocarbons are called as isoprenoid units. And then synthesis of squalene. Squalene consists of 30 carbon units. Then conversion of squalene to cholesterol, it consists of 27 carbon units. So the first step is the first stage is synthesis of HMG-CoA. So here acetyl-CoA, so this happens in 2 steps in fact. Acetyl-CoA will combine with 1 more molecule of acetyl-CoA and this gives rise to acetoacetyl-CoA. Okay. So to this acetoacetyl-CoA, 1 more molecule of acetyl-CoA will combine and this gives rise to HMG-CoA, that is beta-hydroxy beta-methyl glutaryl-CoA. So the conversion of acetyl-CoA to acetyl-CoA is acetoacetyl-CoA is catalyzed by thiolase reaction, and the conversion of acetoacetyl-CoA to beta-hydroxy beta-methyl glutaryl-CoA is catalyzed by HMG-CoA synthase. Okay. So the next step is in the stage. The stage 2 is a formation of mevalonate. Okay. So a 6 carbon containing unit. So HMG-CoA contains how many carbons? 1, 2, 3, 4 and 5 carbons. Okay. 4, 5, 6. So this has to be converted to mevalonate. So here it is a simple step. It is only we are adding 2 hydrogens to the carbonyl carbon of HMG-CoA. Okay, so that is HMG-CoA reductase will reduce beta-hydroxy, that is HMG-CoA to mevalonate. So the only thing is we have removed COA-SH and we have added 1 hydrogen. Okay, that is coenzyme A is removed and we have added 1 hydrogen. So you have a 6 carbon containing mevalonate over here. So one very important aspect of this step is this is a rate limiting step. This regulates cholesterol metabolism. This controls, this can speed up or slow down the cholesterol metabolism. Okay. So the third stage is production of isoprenoid units. Okay, so isoprenoid unit is an 5 carbon structural unit. So that is characteristic of terpenes. There are two important isoprenoid units, that is dimethyl allyl phosphate and isopentenyl pyrophosphate. Okay, biologically, so these are very two important isoprenoid units. So here, how these two things are synthesized. So the first thing is mevalonate will combine. So mevalonate is converted to 5-phosphomevalonate, that is phosphorylation reaction. So here mevalonate 5-phosphotransferase enzyme will catalyze this reaction. A phosphate group is added. So an hydrogen of hydroxyl group is replaced with the phosphate group, and this phosphate group is provided by ATP. Here ATP is converted to ADP. In the next step, 5-phosphomevalonate is phosphorylated again. It takes up one more phosphate from ATP. One more molecule of ATP is converted to ADP. So the reaction is catalyzed by phosphomevalonate kinase, and here it will form 5-pyrophosphomevalonate. Okay. So the 5-pyrophosphomevalonate, again it will take up one more molecule of phosphate, but this time it will replace one more hydrogen of the hydroxyl group that is present on the third carbon. So it will form 3-phospho, 5-pyrophospho, 3-phospho, 5-pyrophosphomevalonate. Okay. So in the next step, this 3-phospho, 5-pyrophosphomevalonate will lose a molecule of carbon dioxide. So a decarboxylase enzyme, that is 5-phosphomevalonate decarboxylase enzyme, will remove one carbon dioxide. Along with that, one inorganic phosphate, one phosphate is removed from the third carbon atom, and a double bond is created over here. Okay, this carbon dioxide is eliminated and this phosphate group is eliminated. Okay, so a double bond is created. So here you got an isopentenyl pyrophosphate. So it contains 5 carbon unit and an iso group over here. So this is called as isopentenyl group over here, and so one isoprene unit. So now we have synthesized one isoprene unit. There is one more isoprenoid molecule. So that is an isomer of this delta 3 isopentenyl pyrophosphate. Why we call it as delta 3? Because double bond is at the third carbon atom, 1, 2 and 3, correct? So this delta 3 isopentenyl pyrophosphate is isomerized to dimethyl allyl pyrophosphate. So this carbon dioxide is not there over here. I should have removed somehow. I'll correct it after your class. Okay, so ignore this. So it should be here. It should be only here. Okay. So here the only difference is in the isomerase reaction. So the double bond is shifted to the second carbon atom. Okay. So this gets converted to CH3, this gets converted to CH, and a double bond is created over here. So the dimethyl allyl pyrophosphate. So these two molecules now build the remaining part of cholesterol molecule, mainly that is lanosterol. Lanosterol can be further converted into cholesterol. Okay. So the fourth step is the synthesis of squalene. Okay, squalene. So here the isoprenoid units will now start joining and make bigger, larger molecules. So a delta 3 isopentenyl pyrophosphate will combine with dimethyl allyl phosphate. So 5 carbon unit plus 5 carbon unit. So the reaction is catalyzed by cis-prenyl transferase. So this will form geranyl pyrophosphate. It is having 10 carbon containing molecule, a combination of 2 isoprenoid units. So here you have 5 carbons from delta 3 isoprenyl pyrophosphate. So this part of the molecule will join with this part of the molecule. Okay, so here 2 inorganic pyrophosphates. So these PPIs are removed in the form of like inorganic pyrophosphate. So and the remaining 1, 2, 3, 4 and 5. So this is contributed by dimethyl allyl pyrophosphate. Okay. So here you have geranyl pyrophosphate, 10 carbon containing unit, and this geranyl pyrophosphate again it will combine with one more molecule of dimethyl allyl pyrophosphate. So again the same reaction. So this will join over here. So cis-prenyl transferase, one more PPI is removed. So this will form farnesyl pyrophosphate. It will form farnesyl pyrophosphate. It should contain 15 carbon unit, 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So this is from geranyl pyrophosphate, and this will add over here, sorry, and it will form dimethyl allyl phosphate is here, 1, 2, 3, 4 and 5. So here, so 15 carbon unit farnesyl pyrophosphate, it contains 3 isoprene units, 1 unit, 2 and this is the third. Okay, so 3 isoprene units, 5 carbon unit containing an allyl carbon atom. So this farnesyl pyrophosphate will combine with one more molecule of farnesyl pyrophosphate. Okay, so farnesyl pyrophosphate will combine with one more molecule of farnesyl pyrophosphate and it will form squalene. So here 2 pyrophosphates are removed, one from first molecule of farnesyl pyrophosphate, other from second molecule of farnesyl pyrophosphate, and this will end up forming not 15, in fact it is 30 carbon unit squalene. It is 30 carbon unit squalene. Make small correction. Okay. And squalene undergoes hydroxylation and cyclization utilizing oxygen and NADPH and gets converted to lanosterol. So this is the squalene structure. So this will undergo hydroxylation. So here the oxygen group is added at the third carbon atom, 1, 2, 3, third carbon atom, and here it will form that is an oxygen bridge. Okay, so that is epoxide bridge. So due to this epoxide bridge, it is called as squalene 2,3 epoxide. Okay, okay. So in the next step, what happens is squalene 2,3 epoxide will undergo cyclization. Okay, so here this bond will move over here, forming the hydroxyl group at the third carbon atom, and this double bond will cyclize the ring A, and this double bond will cyclize the ring B, and this will cyclize the ring C and this will cyclize the ring D. Okay, so here you have the basic sterol of human beings and mammals, that is the lanosterol. From lanosterol you can convert this molecule into many different sterols. Okay, so here you have lanosterol, and through a passage of 19 reactions it will form cholesterol. So that you need not study, so you can say like through a passage of many reactions it will form cholesterol. If you are interested to know how it converts, how lanosterol is converted to cholesterol, I will post that in your classroom, so you can go through, but that is not necessary from your examination point of view. Okay, so you can learn this pathway starting from acetyl-CoA to lanosterol, and from lanosterol you can directly show the synthesis of cholesterol by multiple step reaction. Okay, so how this cholesterol biosynthesis is regulated. Okay, so phosphorylation. So here major regulatory enzyme is HMG-CoA reductase. HMG-CoA reductase exists in 2 forms. Okay, so that we have seen in your previous class. HMG-CoA reductase exists in 2 forms. One is activated form and another one is inactivated form. When HMG-CoA is phosphorylated, HMG-CoA reductase is phosphorylated, it will be inactivated. So, and the phosphorylation decreases the activity of HMG-CoA. It reduces cholesterol biosynthesis. Okay, and further, and dephosphorylation will activate the HMG-CoA reductase enzyme. So that will promote the activity of that is cholesterol biosynthesis. Glucagon favors formation of inactive form, hence it decreases the rate of cholesterol synthesis. So if it decreases, why? Because glucagon is released when glucose level is low. So at that point of time, so you require acetyl-CoA for energy production, because glucose level will be low, so you require acetyl-CoA for energy production. So it conserves acetyl-CoA and deviates acetyl-CoA towards TCA cycle rather than cholesterol synthesis. Insulin will, that is insulin, so here glucagon will create inactive form of HMG-CoA reductase enzyme, whereas insulin will activate, so it will dephosphorylate the HMG-CoA reductase, because insulin is released at larger amounts when there is higher amount of glucose. Okay, so there is sufficient energy reserve. So in that case, the acetyl-CoA can be diverted for cholesterol biosynthesis. So insulin will favor activation of HMG-CoA reductase and synthesis of cholesterol synthesis, whereas glucagon will favor inactivation of HMG-CoA reductase, and it will slow down the cholesterol biosynthesis. Okay, so when ATP level is very low, cholesterol synthesis will stop completely until the energy level raises. Okay, so this is how our body manages. So acetyl-CoA is the central molecule, and there are so many things, and the major hormones which control the metabolism or where the acetyl-CoA should go, whether towards anabolism or catabolism, is decided by two major hormones. One is insulin and another one is glucagon. Okay, so in summary to today's class, okay, so biosynthesis of cholesterol. So almost all the tissues of the body participate in cholesterol biosynthesis. The largest contribution is made by liver, intestines, skin, adrenal cortex, and reproductive tissue. The enzymes involved in cholesterol synthesis are found in the cytoplasm and microsomal fractions of the cell. So the site of biosynthesis of cholesterol is cytoplasm. Okay, so this is an anabolic pathway. So it starts from acetyl-CoA. So acetate that is provided of acetyl-CoA provides all the carbons in cholesterol. So the reducing equivalents are supplied by NADPH, while ATP provides the energy. So for the production of 1 mole of cholesterol, so around 18 moles of acetyl-CoA is utilized, along with the 36 moles of ATP is also utilized, and 16 moles of NADPH is also required. Okay, so huge amount of energy is required to synthesize 1 molecule of cholesterol. That is, if you calculate 36 into 18, how much it will be? 36 into 18. Can you calculate 36 into 18? So let me do it. Calculator. Can anyone answer 36 into 18?
648.
Huh?
648.
648, and NADPH, that is 16 moles, correct? So around 16 into 3, it is around 48. 648 around 734, nearly around 730 to 35 ATP molecules are required, correct? Equivalent to that of energy, equivalent to that of 735 ATPs is required to synthesize 1 molecule of cholesterol. Okay, so that is the thing here. So in summary to the overall pathway, so there are 5 stages. The first stage is synthesis of HMG-CoA. Second stage is formation of mevalonate. Third stage is production of isoprenoid units. Fourth stage is synthesis of squalene. Fifth stage is conversion of squalene to cholesterol. Okay, so here you have the first stage, acetyl-CoA. 2 molecules of acetyl-CoA will combine to form acetoacetyl-CoA. The reaction is catalyzed by thiolase. One COSH molecule is released. Then the HMG-CoA synthase will add 1 more molecule of acetyl-CoA to acetoacetyl-CoA, and it will form HMG-CoA. And this HMG-CoA, by the action of HMG-CoA reductase, it will add hydrogen over here. It will add a hydrogen over here and create the alcoholic group, and COASH coenzyme is released. So here it requires 2 molecules of NADPH is converted to NADP. So HMG-CoA is converted to mevalonate. In the third stage, isoprenoid units, delta 3 isopentenyl pyrophosphate and dimethyl allyl pyrophosphate is formed. That is 5 carbon units are formed. Okay, mevalonate is converted to 5-phosphomevalonate, and 5-phosphomevalonate is converted to 5-pyrophosphomevalonate. 5-pyrophosphomevalonate is converted to 3-phospho-5-pyrophosphomevalonate. So here in each step, 1 molecule of ATP will add 1,1-phosphate group. Okay, so and this 3-phospho-5-pyrophosphomevalonate is converted to isoprenyl pyrophosphate by removing carbon dioxide and 1 phosphate group, and then this gets isomerized. Only there is shifting of double bond from the first carbon to second carbon. So it will form dimethyl allyl pyrophosphate. Okay, so then these 2 isoprenoid units will combine. Dimethyl delta 3 isopentenyl pyrophosphate and dimethyl allyl pyrophosphate will combine to form 10 carbon unit geranyl pyrophosphate. Again this will combine with 1 more 5 carbon unit dimethyl allyl pyrophosphate, and it will form farnesyl pyrophosphate. So here it is containing 15 carbon unit, that is 3 isoprenyl units, and 1 more 15 carbon unit farnesyl pyrophosphate will combine, and it will form squalene. So it contains 6 isoprenoid units. So this squalene will undergoes oxidation, then cyclization to form lanosterol. Lanosterol, through a series of 19 steps, it will form cholesterol. Okay, so here, so this as an overall thing looks very complicated, but I suggest you practice it stage wise. Today you practice stage 1 5 times, tomorrow you practice stage 2 5 times, practice stage 1 once, then stage 3, then stage 4, then stage 5. Take this as a combination of 5 pathways, then combine, and then you practice. Definitely you can learn this. This is not that difficult. It is something like permutation and combination. You need to go on adding the molecules. You should